Okay, well, good afternoon, everyone. So just a couple of housekeeping things before we continue on with our lecture here this afternoon. So I wanted to remind you that our second term quiz is taking place, of course, next Monday and Tuesday. So log in at some point in that 48-hour span to complete the quiz. And then, of course, the Wednesday, Thursday, Friday will be our 3-day time to kind of think about rebuttals. So that's kind of the order of things there. Yeah, and then I guess the only other thing I was going to mention here was that hopefully over the next 2 days, we'll finish up this lecture on organization and cells of the nervous system. So I'll try to post the notes for our next topic, which is the electrophysiology stuff. Sometime over the weekend so that you have it ready to go for Tuesday. Questions about anything before we start with our lecture here? Everybody's all good? Okay. So on Tuesday, we ended on this slide basically talking about the three major regions of the neuron. So just as a way to get back into things here, one of the regions of the neuron we said is the dendritic zone. And the function of the dendritic zone is to receive sensory input. And we highlighted that the dendritic zone is comprised of the cell body and also the dendrites. And we basically said that, look, the cell body is receptive to sensory input. And the dendrites are literally just extensions of the surface area of the cell body to allow for that neuron to be able to receive a greater amount of sensory input, right? To be able to interact with many more neurons. And so I'll just again highlight here these little green structures in this diagram, which they say are synapses, because we're going to be talking more about synapses in just a moment. But yeah, the synapse, as you know, is the interaction point between two neurons. And so we have all these synapses here within the dendritic zone where other neurons are able to send sensory input to the neuron that is the focus here in the diagram. We then highlighted this axon hillock region because we said this is where integration of all that sensory input occurs. Right, you have all these synapses and all kinds of sensory input is coming into the dendritic zone here, but it needs to be integrated together, right? It's a process we'll talk more about in detail later in the course, but the axon hillock is the site of that sensory input integration. And then we said that that integrated sensory input gets fed forward to the initial segment of the axon, and then if we have got a sufficient level of sensory input here, if we have gotten up to some threshold value of sensory input, then the axon initial segment will generate an action potential, and then that action potential will be conducted down the entire length of the axon, right? So we said the axon's function is to generate and conduct excitation, which is just another way of saying action potentials. And then when we get to towards the end of the axon, we enter what's called the telodendritic zone, where the axon suddenly branches. So you get all these terminal branches, again allowing for this one neuron to be able to, in turn, stimulate many other neurons or muscle cells or glands within the body. And here, the telodendritic zone is all about transmitting output, right? So sending out signals that can stimulate other neurons or muscles or whatever. And most often, that output takes the form of neurotransmitters, right? Chemical agents that are going to kind of cross the synaptic cleft and act as the stimulus for the next neuron or muscle or gland cell here in the sequence. And much later in the course, we're going to be talking quite extensively about synapses and neurotransmitters and all the receptors. And again, that's the detail that is needed for some of the fourth-year nervous physiology courses that we have in our department. So that was kind of the overview that we ended on last week, or our last lecture. And I wanted to pick up here and kind of explore some aspects of the neuronal structure in a bit more detail, beginning with the dendrites. And the particular thing I want to talk about regarding the dendrites are the so-called dendritic spines. So here you have an image of a dendrite here, and you can see along this dendrite there are these little spines that are sticking off of the dendrite. And as they label there in the top spine, spines have a head, right, a little head region here, and they have a little neck region, and they're connected to the dendrite. And so I want to spend a little time here this afternoon talking about what do we know and what do we think we know about these dendritic spines, the role they play in our nervous system. So the very first thing we have to acknowledge about these dendritic spines is they are only ever associated with excitatory synapses, right? So again, synapses are these connections between one neuron and another. And so an excitatory synapse is a synapse where one neuron is increasing the chances that another neuron will generate an action potential, right? It's using some kind of excitatory neurotransmitter to increase the chances that it will cause an action potential to occur in the neighboring neuron, whereas an inhibitory synapse is the opposite. This is where you have this synaptic connection where one neuron is actually decreasing the chances that another neuron is going to generate an action potential. And so again, importantly, as it says here, dendritic spines are only ever associated with excitatory synapses. And that's an important piece of information because any hypotheses that we're going to throw out there to explain the role and the function of these dendritic spines needs to certainly take that fact into account. So there are two hypotheses regarding dendritic spines that we're going to discuss here in the lecture this afternoon. The first hypothesis is that dendritic spines allow neurons to regulate their responsiveness to stimuli. And so here you're looking at a picture, a little drawing. So here is our dendrite down here, and we've got these two dendritic spines that are coming off of this dendrite. And what they highlight for us with the words there is that the heads of these two spines have the same surface area, right? So when they say spines have equal conductances, that's essentially what they're highlighting for us, that these two spines have heads that have the same total surface area, and therefore when we try to excite this dendrite through these two spines are equally able to receive that excitatory stimulus, right? There's an equal amount of surface area there, for example, for the neurotransmitters to interact with and to generate an excitatory response. What differs, of course, between these two spines is not the surface area of their heads, but rather the length and the width of their necks, right? You can see the one on the left has a very kind of long and narrow neck. The one on the right has a very short and wide neck. And this is actually really important because this difference in the neck architecture influences how easily excitatory signals can travel from the head, where they originate, to the actual dendrite. And then, of course, it's from the dendrite that these signals would go to the axon hillock, and from there to the axon initial segment. So what they're showing us here, what they're telling us is that if you have a long, narrow neck, like this guy here, you have a lot of neck resistance, which means that the excitatory signal that's coming in here at the head is going to have a very hard time making its way through this long, narrow neck region to get to the dendrite. Whereas if you have a short and wide neck, like the one on the right here, any excitatory signal that comes in through the head can very easily travel through that short, wide neck to get to the dendrite. So that's what they're illustrating for us down here in the figure. So here they're showing us, so it says EPSP, which is the excitatory postsynaptic potential. So basically just think of that as like how much excitation is occurring. You can see here that in the one on the left that has the really narrow, long neck that's resistant to flow of excitation, notice how the excitation in the spine is very high, right? But the excitation that gets to the dendrite is very low. So you're exciting this spine here, But because of that narrow, long neck, the excitation, a lot of it gets trapped inside the head and can't really make its way through that long, narrow neck to get down to the dendrite. And if it can't get down to the dendrite, it can't get to the axon hillock, it can't get to the axon initial segment, it can't contribute to exciting and causing this neuron to generate an action potential. By contrast, in the one on the right, which has this lower neck resistance, this wide, short neck, you can see how they've excited it to the same level, but that excitation is not building up in the spine. More of it is actually getting through the neck to the dendrite. So the idea here is that even if you were to excite these two spines equally, right, the chances of getting an action potential from this neuron would be a lot higher with the low, the low R neck there, the wide, short neck, because more of the excitation signal can actually get through the spine neck and get to the dendrite, and again, from there to the hillock, from there to the axon initial segment to cause an action potential to occur, right? If you excite this spine even with the same degree of excitation, a lot of that excitation is trapped and can't flow through the neck, and therefore you're less likely to get an action potential. So we're talking about the probability of an action potential. So on the left, low probability of getting an action potential. On the right, a high probability. Okay? Yeah, it's a probability that we're talking about. Okay, so what we can see then is that by having more or less spines, right, you have more or less opportunity for excitation, but also just by changing the spine architecture, especially the neck architecture, you can have the same amount of sensory input, but again, it could differ in terms of how much of that sensory input is actually being, passing through to the axons, how much of that is passing through to other neurons then, and really how responsive the nervous system can be to that excitation. A little bit of empirical data to kind of substantiate this idea that we can regulate the nervous system responsiveness to stimuli through changes in, say, the number or the architecture of these spines. This was a study where they were looking at mice, and as you probably know, mice have whiskers, right? And these whiskers are kind of tactile structures. They allow for mice to detect, physical things in their environment, detect vibration and air currents and all these kind of tactile stimuli. What they did in this study is they had two groups of mice. One group was the control, so they did nothing to it, but the other group, they trimmed their whiskers to be half the length. And they did this for a 2-week period. They kept trimming the whiskers so they never got longer than half the normal length. So what they were trying to do by this whisker trimming was they were trying to decrease, right, the tactile sensory input into these mice, right? Reduce their capacity to detect tactile information in their environment. And then after the 2 weeks, they were able to look at the spines within the nervous system of these mice, and they were able to see both how these spines were being eliminated and how they were being formed, especially within the regions of the brain that are responsible for kind of perception of this tactile information. You'll notice that the formation was about the same in these two groups, but where they saw a major difference was in terms of the elimination. They saw that the elimination of spines was dropped in half in the mice whose whiskers were being trimmed. And so consistent with this hypothesis, this idea that basically the more spines you have and or the kind of shorter and wider their necks are, the more ability to load excitation into dendrites you're going to have. Consistent with that here, we see that if we reduce the tactile input of these mice whiskers by trimming them up, the brain compensates by doubling the number of, or I guess I should say by halving the number of spines that it gets rid of in order to have more spines in those mice whose whiskers are being trimmed, right? To make the brain tactile regions more responsive to stimuli to compensate for, to offset the reduction in tactile sensory input coming from the whiskers themselves, right? So I'm hoping what you can see there is how there can be this manipulation of spine density and or the manipulation of the spine architecture in ways that can ensure there's kind of this constant sensory input coming into the brain, even as we are reducing the tactile detection capacity of the whiskers themselves. We can see this in the other direction too. I won't show you any data for the sake of time, but it can work the other way too, whereas if you could imagine, I guess, like lengthening the whiskers somehow, if you could double their length, you'd see the opposite, right? You would see spines getting eliminated a lot more, not forming so much. It goes both ways. And especially when we think about going that other way, where you're like increasing the incoming stimuli to the brain, the reason we want to kind of slow down how much of that excitation can actually travel through the neurons is because overexcitation of neurons can actually lead to their death. We're going to see that more clearly later in the course when we start talking about neurotransmitters. I'll get into that a little bit more. But yeah, if you have too much stimuli coming into a neuron, it can actually lead to the death of that neuron. So reducing your spine number, making your necks more long and narrow when there's too much stimulation coming into the nervous system can actually be a protective mechanism to limit that neuronal cell death from overexcitation. The other hypothesis that's been put out there to talk about dendritic spines is it's been argued that dendritic spines are important for learning and memory because they help to strengthen synaptic connections. Now, I don't pretend to be a neuroscientist. I'm not on the forefront of learning and memory and what goes on in the brain. But generally speaking, the general idea is that when we learn something or we are remembering a fact or something, that information is in some way, stored or encoded, in a synapse. And the stronger that synaptic connection is, the better we are able to retain that piece of knowledge, to retain that memory. And so a little bit of data in support of this idea. This comes from a study, obviously in mice, and they were looking at a region of the brain, the hippocampus, that has a well-established role in learning and memory. And what they did is they subjected some of these mice to stress. And what they observed, and you can see it just from the images, but they've also quantified it here, you can see how stressing these mice out, I think with a chronic stress for a long period of time, it reduced the number of spines that were observed on the dendrites in the hippocampus, right? So you can kind of visually see that there aren't as many spines in the stress case as there are in the control, but you can see it here, a statistically significant reduction in the number of spines observed in the stressed mice compared to the control mice. Now, what they did in the same study is they then used that same technique we saw on Tuesday, that same memory test in mice that we saw Tuesday, where it's like we show you the two familiar objects, and then we show you one familiar and one new object later, and we say, well, how much time do you spend with the new object compared to the familiar one? That's a common test of memory in mice. So when they show us here the novel to familiar ratio, again, the higher that value is, the better a mouse's memory is. And then here, they're correlating it with the number of spines observed, and notice this really striking positive correlation. The more spines you have on your dendrites, the better your memory was. And so, again, this hypothesis that these dendritic spines are necessary for learning and memory, that they strengthen synaptic connections necessary for us to retain knowledge and remember facts, we see that playing out here in this mouse study. That's kind of a nice thing because it reinforces the hypothesis, but at the same time, I think when I think about this study too long, I get a little bit nervous because I think about my life and all the stress that I'm under all the time, it seems, for one reason or another. And this study is telling me that that stress is limiting my dendritic spine numbers in ways that are actually compromising my ability to learn and remember things. So yeah, I mean, there's a couple of times now over the last few lectures where we've really been seeing how stress can be a huge problem neurologically speaking. And not to stress you all out, but if we don't figure out how to better mitigate and manage the stress in our lives, we're just basically getting dumber and dumber as a result. I mean, think about your true cognitive potential could really be if you didn't have to worry about these negative impacts of stress in your life all the time. So anyways, if any of you figure out how to make our lives less stressful, I would love to hear about it, and we'd all benefit greatly from it. Okay. The next thing I want to talk a little bit about here, still talking about dendritic spines, is how the number of spines we have within our brain actually changes over the course of our lifetime. So here we have a figure, it's plotting obviously time on the x-axis here, so basically starting from conception, I suppose, at this point here, and then extending all the way out until we die. And then on the y-axis, plotting out the dendritic spine number. And what you'll notice here, of course, is that while you are in utero, so during gestation, we see this rise in dendritic spine numbers, and then that appears to continue even after birth into early childhood. But we eventually get to a point in life where, if we're just following this normal curve here, the black line, we eventually get to peak dendritic spine number, and then after that we actually start to see the number come down, and so they use the term synaptic elimination here. Another very common term used is synaptic pruning, or synaptic or dendritic spine pruning. We actually start to reduce the number of dendritic spines over time, especially during kind of late childhood, early adolescence. And then you can see where the number kind of levels off during adulthood, and then it starts to kind of decline as we get a little bit older, you know, maybe 50, 60, 70. Not surprising too, that's where we start to see more memory and learning degradation. So that's kind of the normal pattern, right? That there is this rise in spine numbers during gestation and early childhood. It peaks somewhere in childhood, kind of decreases and then levels off, and then we get that kind of age-related decline at the end. Now this figure, I want to show you a little bit more detailed figure here. This is one that came from a pretty recent review paper, because they were looking at the number of, well, they call it here synapse density, but it's akin to synaptic, or akin to dendritic spine numbers here. They're showing us basically the number of dendritic spines as a function of age, but you'll notice that they've not put age in years here, they've put age in gestational periods. So whatever the gestational period is, which is how long an animal develops in utero, they're plotting the data here in that unit of time. And what they realized when they did this for the 9 or so different mammals that are plotted up here, they noticed that for every one of these mammals, the kind of peak in their dendritic spine number, and then you start declining thereafter, was always one gestational period after birth, right? So that's interesting because, of course, this figure here makes it seem like we kind of hit our peak dendritic spine number, just before we hit, puberty or something. But it's actually happening way back here, right? The human case, we have a gestational period of 9 months, right? About 280 days. So the study here suggested that in humans, right around that 9-month post-birth period is when we start to see the decline, the pruning of our dendritic spines begin to occur. And it occurs a lot faster. If you look at mice and rats that only have less than a 1-month gestation, they still start to experience their pruning effect, 1 gestational period after birth, less than a month after their birth. So that's interesting, right? We basically are saying that we're building up our dendritic spine number as we are developing, and for the first year of our life, basically, and then we hit that 1-year mark, and then we start to prune out and eliminate these dendritic spines, and eventually get to some level number that persists through most of adulthood, and then a little bit of aging decline at the end. Now, what I want to think about next here is, well, what is the significance of this whole, pruning process? Like, what's the point of having all these spines that we formed during gestation and early childhood, and then we kind of cut them away starting a year or two after birth? What is the purpose of that pruning? Well, one way to get a handle on that is to note from this figure that if you look at individuals who have autism spectrum disorder, ASD, you'll notice that one of the things that characterizes ASD is an inefficient pruning, right? So they end up with way more dendritic spines than is normally the case. And if you think a little bit about, again, this is not, I'm oversimplifying autism, and I understand that, and I don't need you to get too worked up about it, but one of the things that is a common characteristic in individuals with autism spectrum disorder is they do commonly experience kind of sensory overload issues, right? Where they feel like there's just too much sort of input coming into their brains, and they may, as kind of depicted here in this picture, try to shut out some of that sensory information because it's just too much to handle at a given time. What's interesting is if we think about that sort of characteristic, that aspect of autism, and we put it together with this idea that we know that in autism there's a lack of dendritic spine pruning, that has led to the idea that this pruning process that we normally undergo, it may be a way for us to get our brain to focus on the most important sensory and motor functions that we need to do, get us to focus on the most important pieces of knowledge that we need to understand, because we can't handle having everything, right? Our brains can't handle having every piece of sensory information in our environment being collected and processed all at the same time. That leads to so much noise within our brains that we can't really focus on anything in particular. So the pruning process, right? Again, going back to this idea that we can kind of regulate the sensory input into our nervous system through controlling the number, say, of spines that we have. If we prune out some of these spines over time, we can kind of make sure that our brain is able to focus on whatever pieces of sensory information are most important. And this is likely tailored very much to the environment that you're developing in. Right? One of the things I remember when my kids were first born is how everyone around you emphasizes to you just how important those first couple years of life are in terms of brain development. And this kind of jives with that your brain during the sort of second, third, fourth year of life, once you've gotten over that 1 year, that one gestational period hump, your brain is starting to prune away dendritic spines, right? And so presumably it's going to start not just doing this at random, it's going to prune away spines that seem like they're the least important. And presumably that's going to be based on, well, what is the environment that this brain is developing in, right? What kind of sensory information, what kind of knowledge is being passed to it? Is it having to use on a daily basis? You're going to prune based on that input. And so, the thinking here is that this pruning process is it is a normal process, for sure. It may be a way of trying to get the brain tailored to focus on the sensory information that is most important for the environment in which that brain is developing. And if you can't prune properly, then you end up with these sensory overload situations that we see, for example, in ASD and also some other common neurological disorders. Sure. I don't think it means that it's an environmental at all. I mean, as we've been hearing more and more in the news, nobody really has a great handle on what is the underlying cause of ASD. So yeah, I would not misconstrue my comments to mean that ASD is an environmental phenomenon. It just means that the neurological pruning mechanism for some reason is not as efficient in individuals with ASD. Whether that has an environmental or a genetic underpinning, I have no idea. Okay, yeah. The other thing that's interesting when you look at this figure, it also talks about patients who have schizophrenia. And so that's the green line here, schizophrenia. And you'll notice that it's the opposite of ASD. There's actually an over-pruning that happens in schizophrenia. And that's interesting too, because it has been proposed that this could explain some of the characteristics of such patients. So just a little bit of a figure here kind of highlighting some of the symptoms of patients with schizophrenia. And you know, for example, they very commonly have memory issues. Okay, well that can be explained by this over-aggressive pruning of spines for sure. They have impaired sensory perception. Again, that's consistent with this over-pruning and with the hypotheses we've talked about. The other that are interesting here is this idea of hallucinations and delusions. And I think we can also explain this as well, because if you are overly pruning the synaptic spines, and therefore you're getting kind of less sensory input essentially being, coming into the nervous system and less sensory input being processed by the brain, it's kind of like when we were talking about the immune system. You know, an immune system that isn't challenged with things to do starts to just sort of make up its own things to do. Oh, hell, I'll attack this pollen grain that came in, and now you're allergic to pollen. It could be kind of a similar thing here, where a nervous system that is not receiving adequate stimulation starts to get bored and says, well, I'll just make up my own stimuli then, right? And hence you get hallucinations and delusions, where you're seeing things or hearing things that aren't actually there, because your nervous system, in the absence of a normal level of sensory input, might be trying to compensate and just basically create its own perceptions and sensations. And again, this could be explained by this over-aggressive pruning, leading to less sensory input coming into the nervous system, and the nervous system essentially compensating by creating its own sensations and perceptions. I'll also lastly point out here, so AD is Alzheimer's disease. And here what we see is a really significant loss of spines later in life. And so again, this jives very much with the sort of memory impairment that's so characteristic of Alzheimer's patients. Okay, that's what I said. Good. Okay, let's come back to our figure here. There's another part of the neuron I want to explore now. So I want to look in the cell bodies, and in the drawing here, you see in the cell body all these little kind of dark black structures they've drawn in here. And now I've put a label on them. These are called Nissl bodies. Why are they called Nissl bodies? Well, they're called Nissl bodies because when you stain them using this stain called Nissl stain, which is also known by its chemical name, which is cressyl violet, these structures become very visible. So all this purple violet staining you see here in the cell body, so there's our nucleus in the center of the cell body, all these purple structures you see in the cell body around the nucleus, they become very easy to spot when you have stained that nervous tissue with Nissl stain or cressyl violet. Now, Nissl stain is called Nissl stain because it was developed by Franz Nissl. So he was an early kind of neurological histologist trying to study nervous tissue at the microscopic level. And he basically developed this Nissl stain, the use of this cressyl violet, to be able to visualize, and doing so was able to visualize these structures. Now, what it notes here is that cressyl violet really strongly binds to RNA. It can bind to DNA a little bit. I have it up there, but it's not very strong. I went back and bolded the RNA. Like it really binds well to RNA, not so well to DNA. And you can kind of tell that because if you look at the nucleus here, I mean, the only part of the nucleus that's actually got any staining here is the nucleolus, right, where you have lots of ribosomal RNA. The whole rest of the nucleus, with all of its DNA, is not staining here. So, cressyl violet is really more of an RNA stain with a little capability to bind to DNA. But of course, this leads to a question then. You've got these purple structures here, right, that are binding to this Nissl stain. And we know that Nissl stain binds tightly to RNA. So then the question becomes, well, what the hell are these things? They're obviously these RNA-rich structures in the cell body, but what are they? The answer to what they were didn't come around until the electron micrograph was invented. So kind of in the 1950s, when people started examining neurons with the electron microscope, this is what the Nissl bodies looked like under the electron microscope. So what are they? Well, they're rough endoplasmic reticulum, right? And of course, rough endoplasmic reticulum, what makes it rough is the ribosomes that are on its surface, and ribosomes, again, partly comprised of ribosomal RNA. So we have a tremendous amount of rough endoplasmic reticulum present within the cell body of our neurons. This is not the first time that we've seen the rough endoplasmic reticulum be kind of the main part of a cell. This is maybe a good review question here. What was the other cell that we saw in this course earlier where we said, hey, there's an extensive amount of rough endoplasmic reticulum in this cell? Anybody remember that cell? Then it dies after a little while because it's so busy making proteins. And it just dies, and we said that's actually not a bad thing. Anybody remember this? It was a plasma cell, which makes antibodies. Perfect, yeah. So this is the second time we've seen a cell with a tremendously large amount of RER. We said back in the time when they were making, the plasma cells and they have this RER and they're pumping out antibodies, we said the RER is responsible for synthesizing secretory proteins. It's also responsible for producing membrane proteins, right? Because ultimately the same mechanism that you use to secrete proteins is the same mechanism that you use to embed proteins into the cell membrane. And so that's why we have such an abundance here of rough ER in the cell body of neurons. It's because they're making tons of membrane proteins. We're going to learn next week when we tackle electrophysiology, we're going to learn that there are a ton of membrane proteins that are critical to the functioning of neurons. Ion channels and neurotransmitter receptors and all these things. So they're all having their origin in these Nissl bodies, right? These rough endoplasmic reticulum here in the cell body. And so, as it says here, the cell body is really the biosynthetic center of the neuron. But then what we have to think about is, well, a lot of these membrane proteins that are being made by the rough ER, these membrane proteins have to be able to be embedded into the membrane of the axons, the axon terminals, the dendrites. So how do we transport all these things from the Nissl bodies in the cell body? How do we transport them out to these other cellular regions? Well, that leads us into the next thing we're going to talk about here, which is intracellular. Guys, trying to give a lecture here, okay? All right, so yeah, we're going to talk next here about intracellular transport. So how do we move, structures through the cell? And this applies not just to neurons. I mean, it applies really to every cell that you can imagine in the human body. We're just talking about it here in a bit of a nervous system context. So how do we transport stuff from, say, the cell body out towards, say, the periphery of the cell, down the axon to the terminals, out towards the dendrites? And also, how do we transport stuff back if necessary? How do we get stuff from the axon back to the cell body if necessary? Well, we're going to use what's called the microtubular network. So you can see here at the bottom of this figure, we've got a microtubule here. And the way that I like to think about microtubules is that they are basically akin to train tracks, right? So partly I like that analogy because, I grew up in a household. My dad was a railroader. He was a track maintainer from the time I was born till the time that he retired at 70. And they kind of like forced him out. They're like, you're too old to be a railroader anymore. Get out of here and retire. And he did. But so I have this real obsession with trains. And so I like the idea of thinking of the microtubule network and the microtubules in particular as being like train tracks. Right? So when you think about, the GO network or something here in Ontario or whatever you want to think about, any train system, the microtubules are the tracks upon which the trains are going to be going around and carrying their cargo. So we have a microtubule kind of rail network in our cells. And then just like we have with a real train network, we have locomotive engines, right, that are pulling the cargo along the track. And so in our cells, we have two locomotive engines that travel along this microtubule network to pull cargo from place to place. One of these locomotives is known as kinesin, so that's the one here in blue. The other is known as dynin, it's the one here in green. So these are the two locomotive engines, if you will, of this microtubule rail network that's transporting things throughout the cell. Now, as it shows here in the figure, kinesin, so you see it's colored in blue, kinesin is responsible for what we call anterograde movement of cellular cargo, which basically means from the center of the cell towards the cell periphery. So kinesin transports stuff out towards the periphery of the cell. Dynin does the opposite. Dynin is responsible for the retrograde movement of cargo. That's the opposite direction, from the periphery of the cell back towards the cell center. So you've got these two different locomotive engines, if you will, the kinesin and the dynin, and they're responsible for transporting cargo to the cell periphery and then from the cell periphery, respectively. Okay? Okay, just to explore this a little bit more, let's talk a little bit about the structure of the microtubules. So microtubules are made up of a protein called tubulin. And so you can see here that there's two isoforms of tubulin. There's the alpha tubulin and there's the beta tubulin. And so these will actually come together to form a tubulin dimer. And then you build your microtubule by kind of putting all these tubulin dimers together to create this hollow tube-like structure. So we call these things microtubules because they are literally, small tubes, right? They are microtubules. So they're comprised of the alpha and beta tubulin, these tubulin dimers. And one thing to note about these microtubules is that they are highly negatively charged. So the tubulin protein is very negatively charged. I'll come back to the significance of that in just a moment. What I want to talk a little bit about next here is how do these two locomotive engine proteins, the kinesin and the dynin, how do they actually travel along the microtubule rail network? And I'll preface what I'm about to say with the fact that if you've ever had a course where you've discussed how myosin kind of interacts with actin, right? And you've talked about that maybe in the context of muscle contraction, it's pretty much the exact same mechanism, right? So myosin and actin, again, myosin is kind of the locomotive protein, if you will, that kind of walks along the actin filament. The dynin and the kinesin are very closely related proteins to myosin. It's just that they're walking along the microtubule network instead, but the underlying mechanism powering their motion is the same. So that's what this little figure in the top right shows us here. So it starts in what they're calling the initial state. So here is your dynin in this case. The figure is specific to dynin, but again, kinesin moves in a pretty much identical way. So you have your dynin here with this little kind of linker arm, if you will, that's currently grasping the microtubule. That's what they're calling the initial state. And then in part B, they show an ATP molecule binding to the dynin. And when ATP binds to the dynin, that causes the dynin to unlink from the microtubule. That's what you're seeing there in B. And then just automatically, that linker arm will kind of lurch forward. So it unlinks when ATP binds and just kind of links, or not links, but automatically moves forward. And then what happens next is the ATP gets hydrolyzed by the dynin, so it gets broken down into ADP and inorganic phosphate, and that of course releases energy. That energy is going to be used to allow for the linker arm to connect back onto the microtubule, but now because it had extended outward, it's linking a little further on, and then it uses the actual ATP power to kind of pull the linker arm back towards the dynin, and so you end up kind of pulling the whole dynon forward, right? So we'll go through that one more time, right? So you have your linker arm bound to the microtubule. The ATP binds, and so the linker arm disconnects. The linker arm reaches forward. The ATP gets hydrolyzed, allowing the linker arm to grab onto the microtubule again, but now at a further forward position. And then the energy of that ATP hydrolysis allows the linker arm to kind of come back, kind of bend back towards itself. It's attached to the microtubule at this point, so as it tries to bend back, it essentially kind of pulls itself forward. And so this is how the dynon is able to walk itself, basically, kind of along that microtubule network, right? So it's powered by ATP is perhaps the key point there. The last thing I'll mention, because it shows up here, and this is the one panel here that is specific to dynon, is that there are these compounds called ciliobrevins. They're synthetic compounds, and they are inhibitors of dynon. So they can be used to inhibit this retrograde transport that dynon accomplishes within the cell. The reason they're called ciliobrevins, if you know the structure of cilia, right? The cilia internal structure is all microtubules, and so ciliobrevins were in part discovered and developed initially kind of as a way to, in a cilia context, I guess is what I'll say. So that's where that term ciliobrevin comes from. But again, ciliobrevins are these synthetic inhibitors of dynon and can be used to stop retrograde intracellular transport within cells. Okay, where am I at here in this? Yeah, okay, let's start this anyways. We might cover it again tomorrow. But one of the things that's interesting about talking about intracellular transport, especially, oh, sorry. Yes, they don't inhibit kinesin at all. They're specific to dynon. Is there a reason why? So, I mean, the suggestion is that, so they're kind of showing where it acts here. It basically, inhibits the ATPase activity of the dynon. The suggestion would be that the ATP binding site in dynon must be a little bit different in its structure from kinesin, so that the ciliobrevins can get into one and knock it out, but they can't fit into the other. Yeah.
So you also mentioned that it was initially developed in the context of cilia, right?
Yeah.
So was it to inhibit cilia?
Yeah, I think it's not, because I guess it doesn't, so in cilia you have dynon, but there are two kinds of dynon. There's one that's involved in just kind of more of a structural role, and there's one that's involved more in the beating. And the ciliobrevins apparently especially target the one that is just the structural one. So I'm not exactly sure what they were developed for per se, but it wasn't to inhibit the beating of the cilia per se. It's more of a structural thing. Yeah. Any other questions? I'm gonna stop there then, because ultimately I'm not gonna get through this slide in 2.5 minutes, so I might as well just stop there, and we'll pick this up tomorrow. We should be able to finish this lecture tomorrow, so then, like I said, Tuesday, we'll be able to start hopefully fresh on electrophysiology. So enjoy the rest of your afternoon, and I'll see you tomorrow.